My name is Samuel Brody. I'm a research assistant at Mass General Brigham in Boston. I would like to thank the Cancer Genomic Consortium for allowing us to share our work in integrated cytogenetic and molecular studies in hematological malignancies. As someone who is getting a crash course in cytogenetics, a definition that has been helpful to me for understanding the complexities of this field is the numerical and structural evaluation of the genome that does not consider the assay, but rather considers the pattern and mechanisms observed irrespective of the tools being used. Cytogenetics, at Brigham at least, is dominated by the analysis of GTG-banded chromosomes, and array-based studies represent secondary or supportive modalities that are introduced in addition to the GTG banding to further characterize the diseases being studied. The question that has been the center of our work, and maybe the field's, has been, can we or do we need to replace karyotyping? And if so, how do we accomplish this? There have been a number of strategies that have been described over the last 5 years or so, often involving combinations of different technologies to address the duality of copy number and structural variation detection, which can't be accomplished by traditional chromosome studies. Ultimately, we are left to ask, what do we do, or what do we need to do to achieve an improved diagnostic yield? And really, is there a better way to do this? Within the Mass General Brigham system, we are very fortunate to have the opportunity to be exposed to and use some molecular-based approaches. We have been able to use anchored multiplex PCR, or AMP-based fusion studies. This is a molecular assay in which cDNA is ligated onto adapters, allowing for the unbiased PCR amplification of a targeted series of individual fusions. We have been employing this technology specifically for its potential use in hematological malignancies. The question we have been asking ourselves is, what is the clinical implication of using targeted RNA sequencing? How does this change, or should this change, our current practices? In an effort to address this question, we have been fortunate to work with very talented people, namely Melissa Zhao and Valentina Nardi. We collected a series of over 1,700 data points. These were individuals which both cytogenetic and AMP fusion-based studies were performed over a 2-year period. This represented almost 500 patients. We went through this data set and removed samples that seemed inappropriate for this study and analysis. This included lymphomas, specimens that were disease negative, or duplicate samples, among other categories. From this list, we were able to boil down the study to 383 individual patients with active disease. And from this list, we were able to do a central review of their genomics and histopathology. From there, patients went through a clinical tiering process, where we tiered all the structural variants for either assays on a Tier 1 to Tier 5 system. Tier 1 being known and well-established variants, which have known diagnostic, prognostic, and clinical significance, and down to a Tier 5, where no structural variant is detected. Essentially, Tier 1 and Tier 5 variants were those we felt were actionable and had the potential to change or shape clinical management. Of the types of hematological malignancies in those 300 individual patients, we had quite a range of myeloid and lymphoid malignancies. We annotated the frequency of structural variants being defined as a subchromosomal alteration, detected by either fusion studies or GTG-banded analysis across these individual disease types. We were happy to see that the frequency of these structural variants largely fit with what we expected based on published literature. To understand the added utility of fusion-based studies against cytogenetic studies, specifically here cytogenetic studies being GTG banding, we plotted the tier that was established by either cytogenetic and by fusion-based studies. What we expected to see, if there was equivalence achieved, would be everyone would follow along this line. In reality, what we found is there seems to be a 10% increased frequency of the total events in our study, where Tier 1 or Tier 2 variant was detected by fusion studies, but was not detected by cytogenetic studies. We went on to study the disease-specific nature of these findings, and what we found was that almost universally across diseases, when both GTG banding and fusion studies were performed, there was increase in the overall percentage of Tier 1 and Tier 2 detected variants by fusion studies. This specifically ranged within disease types with about a 14% increase in AML up to a 30% increase in TAL compared to conventional cytogenetic approaches. The question that we asked was, why are we seeing this difference between fusion studies and cytogenetic analysis? So we went on to perform this analysis. What we found was that 63% of the actual variants we detected by fusion studies that were unreported by GTG banding were driven by the resolution of karyotyping. An example of this being a KMT2A partial tandem duplication. An additional 20% demonstrated cryptic translocations, and 6% demonstrated cryptic insertions. And approximately 13% of our discordant cases were when the karyotyping was believed to be non-representative, i.e. an outgrowth of the non-neoplastic tissue. So again, we asked a question. Where does GTG banding not have much utility added on the disease type? One notable example we came across in this analysis is a patient whose genomics we are calling MGB001. In this particular instance, GTG banding detected a translocation 922, which was consistent with BCR-ABL1 rearrangement. Concurrent FISH studies of both interphase and metaphase confirm this. Interphase FISH studies demonstrated an unusual pattern of only one BCR-ABL fusion, which was certainly atypical. But when we performed reflexive metaphase FISH studies, this fuse signal presented itself on the derivative chromosome 22, which made us anticipate that this would be a functionally active rearrangement. And yet, when we looked at the targeted RNA fusion studies, there was no evidence of a BCR-ABL1 rearrangement, and subsequent targeted P190 and P210 studies were also negative. Conversely, the fusion studies detected a KMT2A partial tandem duplication as the only event. This suddenly represented a problem. When cytogenetic studies and molecular studies are discordant, which do you trust? In this instance, we turn to optical genomic mapping as an unbiased methodology to act as a tiebreaker. OGM, in this case, acts as a way to facilitate genome-wide characterization of structural variants when sequence-level analysis is unavailable. When we performed OGM analysis on MGB001, we detected a 922 translocation, but BCR-ABL1 was 9 megabases upstream of the breakpoint, and BCR was 260 kilobases upstream as well. Ultimately, we presumed that the nature of the rearrangement was such that the FISH probe produced a false pattern. Moreover, we were able to review the whole genome, and we were able to detect the presence of a 25 kilobase KMT2A partial tandem duplication that was detected by the fusion-based studies. Additionally, the variant allele fraction calculation suggested there was a loss of heterozygosity on this loci. Intriguingly, this was not a unique event. In fact, a subsequent patient we identified had a similar presentation, with a complex karyotype and evidence of a possible derivative 922 that again was shown to be a BCR-ABL1 rearrangement. This patient was also more unusual. This patient had AML, and this patient, who was first seen at an outside reputable clinic, replicated the suspicion that there was a concern for a BCR-ABL1 rearrangement. Once again, we turned to OGM, and once again we identified a 922 rearrangement, but the breakpoints didn't influence BCR or ABL1. So the question we have been asking ourselves is, how much do you trust your eyes? Here are 3 patients, each of which have a reported 922 rearrangement, but only one truly has the BCR-ABL1 rearrangement. This concept we ultimately named chromosomal mimicry, a phenomenon we described as when a chromosomal morphology mimics a clinically relevant structural variant, but lacks the true gene-level rearrangement. We observed this pattern in 3 instances in our cohort with a frequency of just below 1%. The frequency of chromosomal mimicry even exceeded the frequency of cryptic insertions. So in summary, while false negative karyotypes have been well described, false positive results also occur. As a potential fix to this, methodologies with gene-level resolution should be strongly considered to verify each structural variant, and that integrated diagnosis can be used to identify these driverless tumors, and may enhance the detection of these false negative and false positive cases. With that, I would like to acknowledge Melissa Zhao and Valentina Nardi again for their contributions to this study, and especially Adrienne Dubec for being an excellent mentor in introducing me to the field of cytogenetics. Thank you.